Welcome to the Fertility Queen podcast. Today we are lucky enough to be joined by Amy Clements, one of our senior scientists, embryologists at Number One Fertility, and she'll be giving you the inside scoop at what happens at Number One Fertility behind the scenes. Amy, introduce yourself and tell us about yourself.
I've been working for Number One Fertility since 2018, just after we opened up. Before that, I'd worked at several clinics in Melbourne and overseas.
And what does it take to become a scientist or an embryologist?
So most embryologists have done their science degree at university. All of our scientists have gone on to do postgraduate qualifications in embryology. So they've studied 5 years or so at uni to get where we are now. And then once you start the job, you do a bit of a 2-year internship in terms of learning the ropes. And then you become a junior scientist and work your way up.
And where are you placed in our lab at the moment?
I'm the team leader.
The team leader at Number One Fertility in Emerald City.
That's correct.
Yeah. And what do you love about your job?
I love the fact that we are giving people something that they may not be able to have otherwise. The ability to help people have a baby is just magic. And when the patients bring their babies in to visit, it is unbelievable because you look at the babies and you think, I met you when you were a couple of cells big. And yeah, it's just extraordinary. It's a perfect balance between sort of science-based and patient contact-based. It's, a job unlike any I've ever done before. So yeah, I really love it.
And what would you say to young listeners thinking about what to do with the rest of their lives? Would you say being an embryologist is a great pathway?
I'm really biased. I think it is one of the best jobs. And I think, like I said, there's so much knowledge that goes into it that you feel like, you're really empowered with what you know. But it's also just the fact that you are really able to help people and help people at face value, I think is really important. So, it's a really lovely job.
And I think it's very important to remember that embryologists are very important in your IVF journey, and you're the people behind the scenes because patients don't often get to see you. They see you on their transfer day, but they don't see all the hard work you're doing in the lab.
Absolutely, and I think one of the things that we try really hard to do here is to make sure that our staff are really empathetic and compassionate because we understand completely that the IVF journey is a huge thing to go through, and we want to make sure that we can support you in whatever ways we can, whether that be with, answering another question or just, being there for you when you have your embryo transfer or your egg collection. I think that's really important because, we are such a huge part of the IVF experience and you want to feel like your gametes and your embryos and your future babies are in really safe hands.
So let's get to the egg collection day where we are planning. It's a very exciting day for the patient. She's worked for months and months to get to the egg collection day. And Dr. Ling comes in. I consent the patient outside theatre. I ask her what time her trigger injection was taken and I make sure she's taken the right injection. And then I know that you talk to the patients outside theatre as well. What's that conversation about?
Yeah, so it's basically just a run through what's going to happen inside the theatre, and we talk about what the patient has consented to in terms of their treatment, answer any questions that they've got, any specific requests that they'd like us to follow up with after and along the road. And then once you're in theatre, the embryologist will ask you for another witness. So we'll get you to tell us your full name, your date of birth and your address. And that's really important that we do that separate from the hospital. So witnessing is such a huge part of what we do in terms of the embryology side of things. Obviously, it's a huge concern for most patients. How do I know that these gametes belong to me? How do I know this is my embryo? So we have dual witnessing system where we have two embryologists. You'll often, if you're having a transfer, you'll hear us call out in the background going, can I have a witness, please? That's where asking a second person to come over, and at that point in time we're focusing on the identification that's on those gametes and making sure everything matches. Complementary to that, we also have an electronic witness system that tags all of your gametes and ensures that, when they're together, that they're the right ones. And it makes a very loud noise if anything doesn't match. So it's a really big thing that we focus on, and it's something that obviously we take very, very seriously, but we completely understand people's concerns when they talk about, is that going to be my eggs and sperms?
And what's the witnessing system called?
It's called RI.
RI witnessing system.
Yeah,
that's great. And that gives patients comfort in knowing that we're using two different techniques to make sure
absolutely
that their gametes, their egg and sperm, belong to each other.
Exactly right.
Yeah. Okay, so we do the egg collection. So you're in just close by to me, and with what equipment do you have at that point?
Yep, so it's really important. Eggs are really temperature sensitive. So it's really important that once they leave the follicle, that fluid's drained through a whole heap of tubing and kept into a warm test tube warmer. And then that comes straight through to the lab where you'll be able to see us from where you're having your egg collection. And then we have a temperature and gas controlled crib that we work out of that keeps the eggs nice and warm and makes sure that they're really stable. And at that point, we're able to look through the follicular fluid to find an egg. So what we're looking for amongst the fluid is basically the eggs are disguised at this point in time once they've been drained from the follicle and they're covered in a cloud, but we're able to tell that there is an egg in there most of the time, and then we take it back with us to the lab.
And what's a cloud? Is that a cloud? It's called Cumulus Cells, is that right?
Correct, and the Cumulus cells provide the nutrients that the egg needs to become mature, and it's sort of like its little nest that it lives in.
Because I know sometimes when I do egg collections, you might yell out to me, there's no cumulus cells, and that might be on a patient that's not getting many eggs. So what would that mean? If there's no, does that mean I've gone into a follicular cyst maybe rather than an actual
Yeah, it could
follicle?
be. And sometimes we can tell that, and you can tell that obviously by the volume that we drain from the egg. The fluid looks different depending on whether or not it's a cyst or whether it's an actual follicle. So we're able to give that feedback to you and let you know what we're seeing, and that can help you guide the patient after the egg collection in terms of what's happened.
And particularly sometimes if the trigger doesn't work, and we do have that happen,
Yep.
it's very rare, but sometimes the trigger injection may not work and you'll get empty sort of fluid at that point, won't you, without any cumulus cells?
Exactly right. And that's, we're able to feed that information back as we go. So there's a lot of communication between Lynn and the embryologist in terms of what we're seeing, how many eggs we're seeing, and then that way at least, you're up to date with what's going on, and we're keeping track on our end.
So we also tell the patients not to wear perfume, and the staff in the theatre. What does perfume do to the eggs?
Yeah, perfume contains VOCs, which is volatile organic compounds, and they are really detrimental to eggs and sperm and embryos. So same for your partner coming into the clinic. We also ask that they refrain from wearing any sort of cologne because the smell can travel quite a distance, and we want to just make sure that everything is in the best environment it possibly can be in.
So you've taken the eggs, so you've taken them back to the lab. What do you do with the eggs then?
Yes, a good question. So it depends on what sort of treatment the patient's having. If it's natural insemination, those eggs are then transferred into a dish. We add 100,000 sperm to those eggs and we just leave them to see how they go. And basically the idea behind that is that the sperm will make their way to finding the eggs. So it's similar to what happens in the fallopian tube. However, we're just helping the eggs and the sperm actually physically be closer together. So we're just cutting out the distance. The sperm should chew through the cloud, and then one of them will hopefully get into the egg and help that egg to fertilise. If we're going down the ICSI pathway, the eggs are taken back to the lab and we take off the cloud that they're surrounded in. And at that point in time, we're able to assess what's called the maturity of the eggs. So not all eggs are going to be mature. The maturity of the egg basically indicates whether or not the egg is ready to receive the sperm. And at that point then, we can start to look at how many eggs we've got that are mature for ICSI, and then we can start to do those injections.
So after we've got the eggs and you're about to do ICSI on them, how do you assess the egg before you do ICSI?
So when we look at the eggs, we're looking for their maturity, and how we can tell that is the presence of a polar body. So once the egg has extruded the polar body, that indicates that it's given away its second set of DNA and it's ready to receive the sperm. Our microscopes are fitted with a polarizer. So that's an additional thing that we do for all cycles here. And basically, when you're looking at the egg, it's assumed that the DNA sits underneath the polar body. In 30% of cases, we know that that's not the case. Sometimes the DNA can be sitting at a different point inside the egg. The polarizer uses light to show us exactly where that DNA is sitting, and that allows us, when we do our injection, to avoid going anywhere near that DNA, and it keeps it nice and safe away from where the sperm goes in.
That's fascinating. So I remember you used to have to take the egg out of that particular microscope and put it into another microscope to do that. Does that happen now? I don't think it does. Is it just a click of a switch?
Yep, exactly right. It's as easy as just pressing a button on the microscope, and the spindle pops up and we're able to see where it is and write down that we've visualised it or not visualised it in some cases. And that's all additional information that we can use to, tell the patient what's going on when we're looking at the eggs.
And that's fascinating. And I know the egg's kept in a chamber still at that point.
Correct, exactly right. So at all points we try and we do keep the eggs at their optimum temperature and ensure that they're gassed, and that just means that we're helping the eggs do the best that they can by providing the optimal environment.
So whilst you're performing the ICSI procedure, is the egg enclosed in a certain environment?
Yeah, so when we have a chamber that sits on top of the ICSI microscope, and that aims to mimic the environment that the egg would otherwise be in the uterus. It's temperature controlled, and it also aims to control things like the CO2 and the oxygen.
Tell us about the spindle of the egg. That's fascinating. Is that where all the chromosomes are held?
Yes, that's correct. Yeah. So the idea is that we don't want to disrupt that when we go in to do the ICSI. We want to just avoid that, so that's why we use the polarized light.
Yeah, so that's great. And we know that from studies that about 30% of the time the spindle's not where it's meant to be. Is that correct?
Yep, that's correct. So we're able to use the light refraction in the dish to exactly see where the spindle is at, and that way we can avoid that entirely when we go to do the injection.
Yeah, and I love my notes from the lab where they comment back to me that the eggs were missing their spindles. So when the egg doesn't fertilise, I can actually say to the patients, well, this is probably why your egg didn't fertilise, that the spindle, which is holding the chromosome, has been disrupted.
Absolutely, and that's research that we've done in-house where we've looked at the spindle, whether or not we've visualised it and whether or not that's associated with a lower level of fertilisation. It's not always the case, but we've noticed an association in terms of the spindle not being visualised and the egg having a slightly less chance of fertilising. So that's feedback we're able to give you, and hopefully that helps flesh out a bit of a picture in terms of what's going on.
Yeah, it is very helpful. And so we've fertilised the egg, we've put them in the incubator. And what sort of incubators do we have at Number One Fertility?
Here at Number One Fertility, we use the EmbryoScope Plus to keep the embryos safe. And what that does is it takes photos of the embryos every 10 minutes, and we're able to track their development as it goes along. So once those eggs have been injected, they get safely put away in the EmbryoScope, and the beginning of that egg's life starts. We take the pictures every 10 minutes, and then we're able to have a look at them over the course of the 6 days that they're in culture with us.
And when does the patient get a phone call from the embryologist?
So once the eggs have been collected at egg collection, they get their sheet of paper that you give them that has the number of eggs collected on it. The following day, we give the patients a call to run through with them how many eggs were collected, how many of those were mature, and we were able to inseminate or inject depending on the treatment type, and then how many of those have fertilised.
And how many eggs would you expect to fertilise?
We tell patients to hope for about half. That's a general number across the board. Obviously, some patients get more than that and some patients get less than that depending on their history.
So once we create the embryo, it goes into the incubator, and on day 3, the embryo is usually around 6 to 8 cells. And what do we do at that point?
So on day 3, we know that the embryo's nutrient requirement changes, and what we want to do is help provide them with their optimum diet, essentially. So on day 3, we do a media change. We use sequential media here. So what we want to do is just top the media up for the embryos to make sure they've got everything they need. So the dish is very briefly removed from the EmbryoScope, and then it's popped into a chamber, which is, again, temperature and gas controlled. The media is changed over, and then it's very safely put back into the EmbryoScope Plus.
And it's very exciting what happens then, because the embryo goes from 8 cells to how many cells on day 4 does the embryo have?
It's a good question. So day 4, we start to look for embryo development to be continuing, but we're also looking for what's called compaction. So basically the embryos have been on autopilot for the first 3 days. So they're using egg DNA to propel themselves along, and they just continue to develop and to divide and make new cells. Basically from day 4, the embryos start to use the embryo DNA, and that is a big pivotal change inside the embryo, and we can see that by the cells starting to fuse together, and that's called compaction. So compaction gives us an indication of which embryos are going to go on to become a blastocyst, because statistically we know that the embryos that are showing us compaction on day 4 are the ones that are more likely to become a blastocyst on day 5 and day 6. So that's the reason why we've changed the model of care that we use here. We don't call you on day 3, we call you on day 4, and that way at least we're able to help manage your expectations of what we think is going to happen in terms of the blastocyst development for your embryos.
That's very interesting that the egg DNA helps that embryo get to day 3. And so then it's the embryo DNA that gets it to day 5. So if someone's got poor egg quality, we may not see that embryo go on to day 3. Is that correct?
Yeah, sometimes we see that unfortunately the embryos sort of run out of puff on day 3. They stop developing and stop dividing. And yeah, we can put that down to the egg DNA, sort of getting them to a point, and then they're unable to make that change over and start to use the embryo DNA.
And that's why I always supplement my patients with a lot of antioxidants, because there is some evidence that antioxidants may improve the mitochondrial function of the egg.
we appreciate anything that you can put into the eggs and the sperm prior to coming to the lab, because once they are with us, we can't do anything to improve the quality of the eggs. But if you can make those lifestyle changes and add those supplements and things like that to your diet, that would be really helpful. And that way it gives you the best chance of making the best possible embryos you can.
What about the mitochondria of the egg? What does that do?
The mitochondria is the powerhouse of the egg. It helps provide the energy that it needs to get on with it.
So after day 4, so it's compacted, and then it goes on to the next stage on day 5 where it's starting to hatch and become a blastocyst.
Yep. So once we've seen the embryo start to compact, it starts to do what we call blastulation, and that's where the embryo starts to form 2 sets of cells, and we can see one of them is called the trophectoderm, which is the part that forms the placenta, and the other part is called the inner cell mass, which is what forms the fetal cells. So that is what defines a blastocyst, and we hope to see those heading towards day 5 and day 6.
And the eggshell, so the eggshell contains the embryo for the first 5 days, and then after that the embryo hatches out just like a chicken hatches out of its shell, so to speak.
Yep, correct. So the embryo will eventually leave the shell that it lives in, much like a chicken leaving the egg. Whether or not it happens in the dish or whether it happens in you, we know that either or is fine. If you have a transfer on day 5 and your embryo is not hatching, that's okay. It's still got a couple more days inside of you in order to get there. And same for day 5 and day 6 culture.
So on the day of the transfer, it is actually a very minor procedure for me to do. I know the patients are very anxious, and that's probably really in the whole scheme of the whole journey of fertility treatment is the most important day. So I'll always do a practice transfer on every single patient that comes into the clinic, and so that I know when I've got that embryo in my hand, I'm going to get that in very easily. So we just do, it's like a pap smear type procedure. So we put the speculum in, and I put the outer catheter into the cervix and make sure I'm ready for the embryo, and then I'll yell out to you. And what do you do with the embryo? Where is the embryo at that point when I'm doing that?
Yep, so the embryo is in a chamber that is, again, gassed and temperature controlled, and we, once you're ready for it, load the embryo up into a very fine catheter, the inner catheter, and we quickly walk that round to you to make sure that its temperature is maintained, and then you thread that through, and we plunge a small, teeny tiny bit of media and a bubble into the uterus.
So often the patients look at the screen and they think that little bubble is their embryo, but you can't see the embryo.
You cannot see the embryo,
Yes, it's just a little bubble.
Yep.
But they do get excited to see that little bubble.
Yeah, so the embryo is microscopic, so it's basically the tiny, same size as smaller than a pinhead, so you can't see it at that point, but the bubble makes for an excellent way to sort of visualise whereabouts the embryo and the fluid have landed inside the uterus.
And I call it the sparkle, usually.
Yes.
So now we get to the embryo transfer day. So everyone's very excited, and we've had Nurse Jen running up and down the corridor, making everyone feel relaxed and very special on their day, and spreading her baby dust that she does. And then we get the patient into the room. So once the patient's in the room, what do we do then?
So the embryologist that morning has chosen the embryo for transfer, and we've got a variety of ways that we choose which is the best embryo. But the main one is we watch the video that the Embryoscope Plus has provided us. We can look at the different milestones that the embryos have made along the way and whether or not it's fitting in with what we'd be hoping it would be doing. Once that embryo is chosen, it's moved into an assigned dish that's specific to each patient. We'll have a chat with Lynn and let her know how the embryo is looking. We do another witness in the room with the patient, and that's just to triple check that it is you. And then once you're ready, you call for us to bring the embryo through, and it's loaded up into a tiny catheter, and we bring it into you.
So how do we choose which embryo is the best embryo for transfer?
So we use the video that the Embryoscope Plus has provided over the course of the 5 days the embryo's been in culture. Complementary to that, we also use the KID score and the Life Whisperer AI in order to build a picture of which embryo is looking the strongest at that point.
And the KID score gives you a score, and the artificial intelligence also gives you a score.
Correct, and then we are able to rank the embryos in accordance with the video as well. And then the video is loaded onto a USB stick, and that's given to the patients to take home and watch at home.
I know the patients love that. They tell me that they watch it over and over again.
The videos are amazing. They're like our favourite part of the job, to be able to see how the embryo started and it was just a simple little egg, and to see how beautiful they are by the end of it. It's quite magical.
Yeah, and it's a great thing for the patients to take home, and particularly when when their baby turns 21, they can show that at their 21st birthday.
Exactly right. We call it like baby's first.
Yeah.
Baby's first video. Yeah.
Yeah, it's great. I know the EmbryoScope Plus is quite fascinating, and I know the embryos go into their own chamber and then they're locked away in their own little position, and they're untouched for the 5 days that they're in that incubator. It's quite amazing. And I sort of look at how they slide around, and it reminds me of the old CD-ROMs where we used to put CDs in and you choose your CD to play. So I know the embryos slide around into their own slot, and they just stay there like their womb for 5 days. So it must be better than the older culture and EmbryoScope systems that we used to have in place, where we used to open the doors, or the scientists used to open the doors, and other embryos were in there. There must be a difference.
Yeah, absolutely. So older clinics will work with incubators where you open the door and you expose all the embryos that are in that culture system to the air and the temperature change. The EmbryoScope Plus is different in that like you said, once the dish that contains all the embryos has gone in, it's locked away and it's very separate from every other dish that goes in there. So anytime we need to pull another patient's embryos out, none of the embryos are disturbed in the process of doing that.
And that must make a better environment for all those embryos.
Absolutely. The continuity is really important, and the same for the video that's recorded along the way. We have continuous film essentially of the embryo's development, and it's really, really interesting to see all the different things that the embryos do compared to static images. So previous clinics I've worked at, you take the dish out, you have a look at it once in the day, and you see what you see at that moment, and then the dish goes away. So essentially you're guessing what the embryo is up to. With our EmbryoScope Plus, we're able to know exactly what that embryo is doing. Is that a real cell? Is that not a real cell? How did the embryo come to be, and why does it look this way? We've got all those answers because the video is there. It's really, really interesting. I think I've learnt more in the 2 years here looking at those videos of the embryos than, I could have ever learnt from static images.
So, Amy, another question I get as a fertility doctor is, what was my embryo graded?
It's a good question. Look, embryo grading shouldn't define your chances of getting pregnant. The embryo grading was designed to be able to tell the embryos apart. So an A embryo, for example, would have slightly more cells than a B or a C grade embryo. But it is not indicative of whether or not the embryo is going to make a baby. I think a lot of people get really hung up on the grading of the embryos, but the reality is all sorts of embryos do make babies. All sorts of grading, does make babies. We have so many other ways of assessing an embryo that we try not to get hung up on the grading. So we use the Life Whisperer AI, we use the kids score, we use the video, and all together that helps us get a really good image of what sort of quality embryo we think it is.
And I know with my patients I do recommend embryo testing. Can you just briefly tell us what embryo testing is?
Basically, we want to get a picture of what the chromosomes inside the embryo are doing, and how we do that is when the embryo reaches a blastocyst stage, the embryos hatch out a few cells, and we're able to take those away without interrupting the rest of the embryo and touching the cells that become the fetus. And those cells are then sent off for testing. And when they come back, the genetic counsellor gives the patient a call to let them know the status of the embryo. We can't create more normal embryos doing it this way, but we can rule out the ones that weren't going to give you a pregnancy and save you that trouble.
And also to save the trouble of having a miscarriage and all the trauma that
Absolutely.
that causes.
Yep. IVF's hard enough, let alone going through something like that as well.
Or getting to 10 weeks and
and
finding your baby's got an abnormality.
Yeah, and a lot of clinics don't believe in the genetic testing, but it's something that we really strongly believe in. And we do it because we want to give you the best chance. We want to get you there as fast as we can in terms of getting that baby in your arms. So, yeah, it is something that we really do encourage.
Great. Thank you, Amy, for joining us today. That was so informative. Is there anything else you would like to tell our listeners?
I think the biggest thing is the embryology team is here for you. If you ever have any questions, you hang up the phone and you think, I don't know what she said, or what number did she say for the number of eggs collected, you just call us back. We're always happy to talk about things further. IVF is so emotionally stressful. It's such a big process that we're here to help you And I think information is power, and the more information that we can give you, the better you'll feel about it, the more confident you'll feel in how the lab's going. And hopefully, we can just get you that baby.
Yeah, and I love that too, Amy, and I love being a doctor in this clinic because it's a team approach.
Yeah.
So we have a team of administration, we have a team of nurses, we have a team of scientists, embryologists, and we have a team of doctors all working to their very best to help you have a baby.
Absolutely. I think that people think that it's all, you're just a number. We don't believe that here. And I think that people don't realise that when the pregnancies come through, we all go, oh, so-and-so's pregnant. It's really exciting. Sometimes we'll go back and we'll look at the embryo and we'll think we did that. Like, it's really, really special. And if you do have a baby, please bring it in. It's the absolute highlight of our week. It's so lovely to meet these humans that we helped create.
Well, thank you, Amy. That's been very informative today, and hope you're enjoying our Fertility Queen podcast. Stay tuned for our next one.
Thanks, Lynn.